Hello all, this is Dr. P. Miriam Puma. Today we'll be studying about amino acids and proteins. So this is an introductory video which is made for the new APSHE curriculum wherein you have applied biology in which a small chapter is there, amino acids and proteins. So this video is all about introduction to the biomolecules, amino acids and proteins. The name protein was suggested by Berzelius, and it was first used by Mulder in the year 1840. This word is derived from the Greek word which means first, and proteins, these are considered to be very complex nitrogenous compounds. These are found in all living forms, and the basic functional unit with which the proteins are made up of are amino acids. We have a total of 20 different types of amino acids. And these amino acids, they consist of an amino group and a carboxyl group. An amino group and a carboxyl group. And two amino acids, they are linked by peptide bond. These are formed between the amino group and the carboxyl group, these two groups. And you present it as CO and H bond. So this is the peptide bond which is formed between two amino acids, and this is the representation of a dipeptide. Amino acids are variously classified based upon the side chain that they have. These could be hydrophilic or polar amino acids or hydrophobic, which is non-polar amino acids. Under hydrophilic amino acids, we have positively charged amino acids, negatively charged amino acids, and uncharged amino acids. While in the case of hydrophobic amino acids, we have amino acids containing aliphatic side chain and aromatic side chain. Examples for negatively charged amino acids are aspartic acid and glutamic acid, while positively charged amino acids we have lysine, arginine and histidine. Serine, threonine, asparagine, glutamine, cysteine and tyrosine are uncharged amino acids. Glycine, alanine, valine, leucine, isoleucine, proline and methionine are aliphatic side chain containing amino acids, while phenylalanine and tryptophan are the examples for aromatic amino acids. Glycine is the first example for an amino acid. So you see that an amino acid basically consists of a chiral carbon, amino group, carboxyl group, a hydrogen, and from here the side chains will be varying from all the amino acids. First let us see the hydrophobic amino acids. Glycine, which is represented with the single term G, and this is the three-word code GLY. The side chain is a simple hydrogen, and glycine is the example for an amino acid which doesn't have any optical activity because there is no chiral carbon atom. Next comes alanine, represented as ALA. It has methyl group substitution, while this is valine, represented by VAL. To this valine add a CH2, you get isoleucine. Just here you place CH2 and you get isoleucine, represented by LEU. Then comes isoleucine, represented by capital I and ILE. Next is the non-polar side chain containing amino acids. So this is methionine, which is a sulfur containing amino acid, represented by M or MET. This is phenylalanine, represented by F or PHE. This is tryptophan, represented by W or TRP. While this is amino acid, which is proline, P or PRO. Next, let us see the hydrophilic, acidic and basic amino acids. So we have under hydrophilic amino acids containing polar side chain, which includes serine, which is a hydroxy group containing amino acid. Serine and threonine are the examples for hydroxy amino acids. So this is serine represented by capital S or SCR. This is threonine capital T or THR. Then this is cysteine again, a sulfur containing amino acid, very important sulfur containing amino acid, abbreviated as capital C or CYS. Here is tyrosine, aromatic amino acid, which is represented by capital Y or TYR. We have asparagine, a basic amino acid, represented by capital N or ASN, while this is glutamine. So these two amino acids, they are amides of the glutamic acid and aspartic acid. Okay, so this is glutamine, abbreviated as capital Q and GLN, asparagine abbreviated with the letter capital N or ASN. So these are the two acidic amino acids. We have aspartic acid and glutamic acid, or their ionic form we call them as aspartate and glutamate, capital D or ESP, while this is capital E or GLU. Next we have the basic amino acids. Examples include the lysine, which has an epsilon amino group represented by capital K or LYS. Then we have arginine represented by capital R or ARG. And finally histidine represented by capital H or HIS. Once again, the classifications, there are various types of classifications. So under hydrophobic amino acids, hydrophobic non-polar amino acids, we have glycine, alanine, valine, cysteine and proline. Again we have leucine, isoleucine, methionine, tryptophan and phenylalanine. While under hydrophobic, we have this serine, threonine, tyrosine, asparagine and glutamine. Under charged amino acids, positively charged ones, which are basic, we have lysine, arginine and histidine. While negatively charged amino acids, we have the two acids, aspartic acid and glutamic acid. Now let us see about the structural organization of proteins. So the organization of proteins, it is studied under four levels. We have the primary structure, secondary structure, tertiary structure and quaternary structure. So what is primary structure of proteins? Here we'll be trying to find out the sequence in which the amino acids are arranged, and we know that amino acids, these are linked by peptide bond, and it's a linear sequence of amino acids like this. They will be linearly arranged. Some structural proteins, they'll be remaining in this linear form only, as in the case of the example is silk fibrion. The silk fibrion protein or silk fibron protein, it consists of glycine, serine as well as alanine, and this is how it is made up of. And these fibers, the individual fibers, they are again linked by hydrogen bonds. So here for silk fibron protein, it is simply the linear structure which is existing. Okay, the linear structure means the amino acids are there and they are linked by peptide bonds. And here the important thing is the sequence in which they are arranged. The sequence in which they are arranged one after the other. This comprises of the primary structure of proteins. Let us see one more example of insulin. Insulin, we all know that it consists of two chains, A and B. So these two chains, they are linked by disulfide bonds. So this is the linear structure. While in the case of chymotrypsin, it consists of 246 amino acids and has 5 disulfide bonds, and it shows a linear unfolded structure, chymotrypsin. The secondary structure basically comprises of the foldings or the coiling and related patterns. This is due to hydrogen bonding or disulfide bond formation. And because of this intra-chain bondings, you'll be able to see that two-dimensional structure attainment. It basically comprises of two structural organizations. One is the alpha helices, while the other one is the beta pleated sheets. In alpha helices, the series of amino acids, you know that they are linked by peptide bonds, and these in turn have hydrogen bonds between the CO and the NH group of amino acids. The alkyl groups of the alpha helices chain, they will not be involved in hydrogen bond formation, but they maintain the alpha helical structure. Every winding turn of an alpha helix, it comprises of 3.6 amino acid residues. That means one winding, it comprises of, from here till here, it comprises of 3.6 amino acids. And you see that the bond is between CO and NH of different amino acids. So here you find the hydrogen bond between this oxygen and the hydrogen. Next is beta strands. Beta strands, it consists of various strands, beta strands, which are linked by hydrogen bonds, and this is between adjacent strands. Okay, so 3 to 10 amino acids, they are combined to create these beta strands like this, beta strand polypeptides, and these beta sheets, they are involved in the formation of these fibrils and protein aggregates, which you see in the case of amyloidosis. So like alpha helices also, here you see that hydrogen bonding will be there between adjacent strands, not within the same strand, but between adjacent strands, and these are separate from each other. Here you see that again this is an anti-parallel beta sheets, while here this is the parallel beta sheets. Anti-parallel and then parallel beta sheets. So you see the pattern of formation in the case of anti-parallel. And this is the parallel one, wherein both the arrangements of these strands is in the same way, they run in the same direction, while these run in opposite direction. So let us summarize the differences between alpha helices and beta sheets. In alpha helices, the amino acids, they'll be present in right-handed coiled rod-like structure, while in the case of beta helices, the amino acids, they are like extended conformation, like you have linear or sheet-like structure. Here you have intramolecular hydrogen bonding within the peptide chain, and because of which you get the spiral structure. While here the beta sheets, you have two strands and it is intermolecular hydrogen bonding. Intra means within, inter means between two strands. Here the pitching is 3.6 amino acid residues for one winding, as we have seen. Here 3 to 10 amino acids, they come together to form a beta strand polypeptide chain. And here alpha helices, they can be a single chain polypeptide, while here uh, it is not a single one. You require two or more beta strands. Here alkyl groups of alpha helices, they'll be present towards the outside of the helix, while here they may be oriented either inside or outside of the sheets. Examples where you find alpha helices are keratin, myoglobin and hemoglobin, while here it is skin fibers or fibria. Then comes the tertiary structure, the third level of organization. Here the proteins, they start folding between the alpha helices and the beta sheets. Here you can find the beta sheet and the alpha helices also. And here you see that as a result of this, you are getting a three-dimensional structure. There may be many types of bonds which are involved in attaining this three-dimensional conformation, which include hydrogen bonding, disulfide bonding, or even ionic bonding. So here, this structure, it's also decided by the hydrophobic interactions that exist between the non-polar amino acids, which will be there. And non-polar amino acids, you know that they'll be water repelling. So they'll be oriented towards the inside of the polypeptide or inside of the protein. So they repel the water, thereby they come towards each other, and they'll be present inside of the three-dimensional conformation. So you have all these interactions which contribute to tertiary structure of proteins. The last of the organization is quaternary structure, which is nothing but the final product which we see. So the polypeptides, they come together to attain this quaternary structure. So obviously you have more than one polypeptide chain involved. So each of the polypeptide chain is differently oriented. So they have their own alpha helices, beta sheets, and the hydrogen bonding will be holding each of the polypeptide. While you see that in the quaternary structure, each of this uh, polypeptide having a quaternary structure, having a tertiary structure, is again linked with each other. And this can be due to hydrogen bonding or disulfide bonds or ionic bonds, hydrophobic interactions, etc. Okay, just like the tertiary structure, you see that these also orient themselves, and all these interactions play a role in attainment of quaternary structure. This is the example of hemoglobin. You know that hemoglobin, it consists of globin chains, 4 globin chains. Okay, each of the globin chain, each chain, it has its own tertiary structural organization, while 4 such chains come together to form the quaternary structure. And this is the heme. And finally, you get the hemoglobin. Globin plus heme, you get the hemoglobin. So let me summarize. You have primary, secondary, tertiary, and quaternary structures. Primary means just the sequence of amino acids, and the bond that is involved is peptide bond. While in the secondary, we have seen alpha helices and beta sheets. Hydrogen bonding is involved. In tertiary structure, you have the 3D shapes because of hydrogen bonding, disulfide bonding, ionic bonding, and hydrophobic interactions. Same interactions play a role in attainment of quaternary structure. It is not just one polypeptide chain, but multiple polypeptide chains will be involved. Thank you for watching this introductory video.